It must have looked to people who were experiencing it as if it was the end of the world and that nobody was going to survive.
October 1347, the port of Messina, Sicily. 12 merchant ships from Crimea pulled into harbor. Port officials walked toward the docks to inspect the cargo. They stopped. Most of the sailors on deck were already dead. The ones still breathing wished they weren't. Their bodies were covered in black, swollen lumps the size of eggs, oozing blood and pus. Blood seeped from their mouths, from their ears, from their eyes. The smell coming off those ships had no name in any language. The officials of Sicily ordered every ship out of the harbor immediately. It was already too late. You have heard this story before. You know how it ends. 50 million people dead, 1/3 of Europe erased, and you were taught where it started. Here is the problem. That part was wrong. In 2022, a team of 40 scientists read the real evidence, not from history books, from the tooth pulp of people who died 684 years ago. Before we go any further, drop your answer in the comments right now. Where do you think the Black Death actually started? China, Mongolia, Central Asia, somewhere else entirely? Put it down below. At the end of this video, we find out who got it right. For 6 centuries, the answer seemed settled. The Black Death began somewhere in the east. It traveled west along the Silk Road, carried by infected fleas riding on black rats aboard merchant caravans and trading ships. It entered Europe through port cities, spread through crowded medieval streets, and in 7 years, from 1347 to 1353, it killed between 30 and 60% of the entire European population. That was the story, and it had names attached to it. Giovanni Boccaccio witnessed the plague firsthand in Florence in 1348. In his collection of stories called the Decameron, he described what he saw with a precision that still disturbs readers today. Swellings in the groin and armpit, black blotches spreading across the skin, death within 3 days of the first symptoms. Boccaccio wrote that the city of Florence lost 100,000 people. He watched his neighbors drag bodies into the streets at dawn because there was no longer anyone left to bury them properly. His account became the foundation, and for centuries, historians built on top of it. In 1976, historian William McNeill published a book called Plagues and Peoples. McNeill argued that the Black Death originated in Central Asia, or possibly China, carried westward by Mongol armies and merchant caravans moving along the Silk Road trade routes. His argument was elegant. It fit the timeline. It connected the dots between East and West in a way that made geographic sense. The problem was that McNeill, like every historian before him, was working without the most important evidence of all. There was no bacterial DNA to test. There were no genomes to sequence. There were no ancient microbes to read. There were only written records, and written records can only tell you what the people writing them understood, which in 1347 was almost nothing about what was actually killing them. The Silk Road theory became textbook. It appeared in encyclopedias, in school curricula, in documentary films, and in almost every popular account of the Black Death written in the 20th century. An elegant hypothesis built before anyone could read the actual biology. And when the biology finally arrived, it told a completely different story. In 2022, a study was published in the journal Nature that stopped historians and microbiologists in the same moment. The research team was led by Johannes Krause at the Max Planck Institute for Evolutionary Anthropology in Germany and Philip Slavin at the University of Stirling in Scotland. The team included 40 researchers from institutions across Europe and Central Asia. What they had found was a cemetery, 2 cemeteries to be precise, in the Chu Valley region of what is now northern Kyrgyzstan. The sites were called Karajigach and Burana. Medieval grave markers at both sites recorded burial dates in 1338 and 1339, with inscriptions in ancient Syriac script that used a specific word for the cause of death, a word scholars translate as referring to a catastrophic epidemic, not ordinary illness. That was 9 years before the ships arrived in Sicily. The team extracted DNA from the tooth pulp of 7 individuals buried at those sites. Tooth pulp is the most reliable source of ancient DNA available to science. The hard enamel surrounding the tooth acts as a protective shell, slowing the degradation that destroys genetic material in other bones. Even after nearly 700 years in the ground, enough readable DNA survived inside those teeth to sequence. What the sequencing revealed was not ambiguous. The bacteria present in those 7 individuals was Yersinia pestis, the bacterium responsible for bubonic plague. And for the first time in history, science was not inferring. Science was reading. The discovery of Yersinia pestis in those teeth was significant. What the phylogenetic analysis revealed was extraordinary. Phylogenetics is the science of building family trees from genetic data. The same way geneticists can trace a human family line through inherited DNA, microbiologists can trace the evolutionary lineage of a bacterium through mutations in its genome. Each generation of bacterial reproduction introduces small changes. Those changes accumulate, and by reading the pattern of those changes, scientists can reconstruct which strain came from which ancestor and in what order. When Krauss and Slavin's team mapped the Kyrgyzstan strain of Yersinia pestis against every other sequenced strain from Black Death sites across Europe and the Middle East, the result was unambiguous. The Kyrgyzstan strain from 1338 was the root node. The direct ancestor. Every strain recovered from European plague victims between 1347 and 1353 branched off from it. The Chu Valley in northern Kyrgyzstan was ground zero. The cemetery itself supported the finding. Of the 467 documented graves at Karadzhigach, the burial rate in 1338 and 1339 was dramatically elevated compared to surrounding years. Something killed a large number of people in that valley at that specific moment. The inscriptions said epidemic. The DNA confirmed what kind. This finding matters beyond the geography. It is the first time in history that the origin of a medieval pandemic has been confirmed not through the interpretation of written records, but through direct genetic evidence recovered from the bodies of the people who died in it. For 600 years, historians pointed to the East in general terms. In 2022, science pointed to a specific valley, in a specific region, in a specific decade, and said, here, this is where it began. With a confirmed origin point and a confirmed bacterial lineage, researchers could now test the existing theories directly. What they found was that several of the most widely accepted explanations did not survive contact with the genetic evidence. The first theory to fall was the China origin hypothesis. McNeill and several scholars who followed him had pointed to Chinese chronicles from the early 14th century as evidence of a major epidemic in China before the westward spread of the Black Death. When researchers examined those chronicles carefully alongside the DNA evidence, the picture changed. The Chinese records described famines, locust plagues, and regional disease outbreaks, but they did not describe the specific pattern of mortality that Yersinia pestis produces, and the genetic lineage from Kyrgyzstan showed no evidence of a Chinese origin branch. The root was Central Asian, not East Asian. The China hypothesis had been built on geographic proximity and historical assumption, not on biological evidence. The second theory that came apart was the single Silk Road pathway model. The traditional explanation described a neat linear journey, Central Asia to the Middle East to Mediterranean ports to Northern Europe. The genetic data showed something more complicated. When researchers compared the mutation patterns across strains recovered from different regions, the evidence suggested multiple simultaneous transmission pathways moving in different directions from the Central Asian origin point. The plague did not march west in a single column. It radiated outward along multiple routes at roughly the same time, which explains why it appeared in so many distant locations within such a compressed time frame. The third assumption that the evidence complicated was the simplest one, that the Black Death was only bubonic plague. A study published in 2023 by researchers at the University of Oslo examined the recorded symptoms described in medieval chronicles and compared them against the known clinical profile of Yersinia pestis infection. Several of the described symptoms, particularly the extraordinary speed of transmission in some documented outbreaks, do not fit cleanly with bubonic plague transmission rates. The researchers proposed that co-infection with other pathogens may have amplified the mortality in certain regions. This hypothesis has not yet been tested with ancient DNA evidence and remains an open question. But it is a question the genetic evidence has made it possible to ask for the first time. Where the Silk Road theory worked from chronicles outward, the DNA evidence works from the bodies inward. The difference in what each method can see is not small. Here is the part of the story that most accounts of the Black Death leave out entirely. The plague did not only kill people, it selected them. In 2022, a separate research team led by Hendrik Poinar at McMaster University in Canada and Luis Barrero at the University of Chicago published a study in Nature analyzing the DNA of individuals buried before, during, and after the Black Death across 4 medieval European cemetery sites. They were not looking at the bacterium, they were looking at the survivors. What they found was a specific genetic variant called ERAP2. ERAP2 is a gene involved in the immune system's ability to identify and respond to bacterial infection. The researchers found that individuals carrying a particular variant of this gene were significantly more likely to appear in burials dated after the plague than in burials dated before it. In other words, people with this variant were more likely to have survived. The estimated survival advantage was approximately 40% higher than for individuals without the variant. 40% in a pandemic that killed 1 in 2 people is not a marginal difference. It is the difference between a family line continuing and disappearing entirely. The Black Death, in the language of biology, was one of the most intense selective pressure events in recorded human history. In 7 years, it filtered the European population through a genetic screen so severe that the people who came out the other side were measurably different, at the genomic level, from the people who went in. Here is what that means in direct terms. If you are watching this video and you have European ancestry, there is a meaningful probability that somewhere in your family line, an ancestor carried the ERAP2 variant that gave them a better chance of surviving the 14th century. The people who built the world that eventually produced you were, in part, selected by a bacterium that emerged from a valley in Kyrgyzstan in 1338. The Black Death did not only reshape the map of medieval Europe, it contributed to shaping the immune architecture of the people who are alive today. The question worth asking at this point is a practical one. How does a scientist read the DNA of a bacterium that has been dead for nearly 7 centuries? The answer begins with the tooth. Tooth pulp is the soft tissue at the center of a tooth, enclosed by dentine and protected by enamel, the hardest biological material in the human body. When a person dies from a systemic bacterial infection like bubonic plague, the bacterium circulates in the bloodstream and deposits genetic material in soft tissues throughout the body, including inside the teeth. After death, the enamel acts as a sealed container, slowing the chemical processes that break down DNA. Bones exposed to soil degrade relatively quickly. Tooth pulp, shielded by enamel, can preserve readable genetic fragments for hundreds or even thousands of years under the right conditions. Thoughtful. The process of reading that material is called ancient DNA sequencing. Scientists extract the material from the tooth pulp, filter out contamination from environmental bacteria and fungi, and then sequence the surviving fragments using next-generation sequencing technology. Because ancient DNA is fragmented, researchers do not read one continuous strand. They read millions of short broken pieces and use computational algorithms to assemble them into a coherent genome. The way you might reconstruct a shredded document by matching the edges of thousands of tiny pieces. The reason this was not possible 50 years ago, or even 20 years ago, comes down to cost and computing power. In 2001, sequencing a single human genome cost approximately $3 billion and took 13 years. By 2022, the same process cost under $1,000 and took a matter of days. That reduction in cost is what made the Kyrgyzstan study possible. The cemetery had been known to archaeologists since the 19th century. The grave markers had been photographed and documented for decades. What was missing was not the evidence. What was missing was a tool cheap enough and powerful enough to read it. The evidence was always in the ground. Science simply had to become capable of listening.
Return to that harbor in Sicily, the 12 ships, the officials who walked toward the dock and then stopped. They had no idea what they were looking at. They could see that people were dying. They could not read the bacterium. They could not trace the lineage. They could not locate the origin. They could only react. And their reaction came too late, and the world changed. For 600 years after that morning, human beings argued about where the catastrophe came from. They had chronicles, they had geographic reasoning, they had educated hypotheses from distinguished historians. What they did not have was the ability to go directly to the source and ask. In 2022, for the first time, someone did. A cemetery in a valley in Kyrgyzstan that had been sitting in the archaeological record since the 1800s finally gave up its answer. Not through interpretation, not through assumption, through the DNA of 7 people who died in 1338 and were buried with a word carved into stone above them that meant something terrible had passed through. The Silk Road theory was not wrong because it was careless. It was wrong because it was built before the tools existed to test it. McNeill worked with what he had. Boccaccio wrote what he witnessed. Every scholar who repeated the China origin story was working from the best available evidence of their time. That is not a failure of scholarship. That is the nature of knowledge before the evidence arrives. What the 2022 study changed is not just the map of where the Black Death started. It changed what kind of question we are now allowed to ask. If the origin of the deadliest pandemic in medieval history can be located precisely in a specific valley, confirmed by genetic evidence from the people who died there, then the history of every major epidemic before the modern era is now potentially readable in the same way. The remains are in the ground, the DNA is in the teeth, and the tools are finally here. 600 years of uncertainty, 7 teeth, 40 scientists, 1 answer. The Black Death started in the Chu Valley of northern Kyrgyzstan, no later than 1338. Everything that came after, the ships in Sicily, the bodies in Florence, the mass graves across Europe, every one of those deaths traces back to that valley, to that year, to a population of people whose grave markers recorded that something catastrophic had arrived. For 600 years, we knew how many people died. In 2022, we finally knew where it came from. And that knowledge revealed something no chronicle ever could. The people who survived were not simply lucky, they were, in part, selected. And the immune architecture that selection produced is still present in the population alive today. The plague ended. The biology it shaped did not. Now go back to your comment from the beginning of this video. Whatever answer you wrote down, take a look at it. A valley in Central Asia that most people cannot locate on a map without help. A cemetery that archaeologists knew about for over 100 years. 7 sets of teeth. and an answer that was waiting in the ground the entire time. The history you were taught was built from the best tools available. The history science is now uncovering is built from the bodies themselves. And somewhere in that difference is the most important lesson the Black Death still has left to teach.